So I'm not an expert in, I guess, in this field, but I would think from a clinician standpoint, it's actually it may be very difficult to sort out, the effect of the hormones, body size, organ size, social activity, lifestyle, how much exercise you do, and whole sort of things that are postnatal. So I wonder if, for the folks who do basic research, have people look at mice that are newborn or mice that are in utero and do RNA sequencing or proteomics of the hearts between male mice versus female mice? Because by the time you look at 8-week-old mice, they've the sex difference has already kicked in. And likewise, for David, you showed the G-tex, but again, these are patients, and by the time they're 40, 50 years old, in which there's a whole bunch of stuff that you just can't sort out the differences. So I just wonder if people have looked at taking newborn pups or just mice that are in utero and just look at male versus female heart. And if the theory of XY has an effect on everything, then you should be able to see a difference, not just for the heart, but for the liver, for the brain, and for other organs, I assume.
We haven't done RNA-seq on in utero, so I can't answer that question, but David had it on one of his slides this morning because it's very clear that sometimes some of these highly penetrant mutations that cause HCM do so in 3-month-old infants. And so that's prior to at least the sex hormone question. I don't know whether anybody else has done sequencing at that level. We have not.
We have not been doing sequencing, but but we have been looking at proteomics, and what we study on a regular basis is the human endothelial, the UVEC cells. And if we look at UVEC cells that reflect just the state of the newborn, we see significant sex differences already in gene expression and also in the response to the genes. And I think this is quite normal in the rat and in the mouse. The gonads start at about 13 at the day 13 in embryonic development to produce sex hormones. And these sex hormones act as DNA methylase, activate DNA methylases or demethylases. And so if you do, and this is what we have been doing, if you study the methylation state of rat and mouse embryos, you start to see differences in the methylation state of cardiac genes already in the embryo. So the biological differences appear very early and they persist a long time during life. We don't know how long, but certainly pretty long.